Lesson 5: Nucleic acids. Structures and functions. The aim of the Lecture 5 is familiarize students with nucleic acids, types of nucleic acids and their functions. Objectives of the lesson: describe the structures and functions of different nucleic acids. Explain the dependence of the Na functions with their structures. Nucleic acids are naturally occurring chemical compounds that serve as the primary information-carrying molecules in cells. They play an especially important role in directing protein synthesis. The two main classes of nucleic acids are deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. DNA is often called the blueprint of life. In simple terms, DNA contains the instructions for making proteins within the cell. DNA is made up of repeating molecules called nucleotides. Why do we study DNA? We study DNA for many reasons. Example: its central importance to all life on Earth. Medical benefits such as cures for diseases. Better food crops. Chromosomes, DNA and genes. Our genes are on our chromosomes. Chromosomes are made up of a chemical called DNA. Genes, units of genetic material that codes for a specific trait. The shape of the molecule. DNA is a very long polymer. The basic shape is like a twisted ladder or zipper. This is called a double helix. DNA nucleotide. A history of DNA. Discovery of the DNA double helix. Frederick Griffith discovers that a factor in diseased bacteria can transform harmless bacteria into deadly bacteria, 1928. Rosalind Franklin X-ray photo of DNA. 1952. Watson and Crick described the DNA molecule from Franklin's X-ray. 1953. Watson and Crick proposed DNA had specific pairing between the nitrogen bases. Adenine, thymine. Cytosine, guanine. DNA was made of two long strands of nucleotides arranged in a specific way called the complementary rule. One strand of DNA. The backbone of the molecule is alternating phosphate and deoxyribose, a sugar, parts. The teeth are nitrogenous bases. For nitrogenous bases, DNA has 4 different bases. Pyrimidines. Cytosine C. Thymine. Purines. Adenine A. Guanine G. Pyrimidines are single ring bases. Purines are double ring bases. Chargaff's rule. Adenine must pair with thymine. Guanine must pair with cytosine. Their amounts in a given DNA molecule will be about the same. Base pairings. Cytosine and guanine always join together CG. Adenine and thymine always join together AT. DNA double helix. Hydrogen bonds. The bases attract each other because of hydrogen bonds. Hydrogen bonds are weak, but there are millions and millions of them in a single molecule of DNA. The bonds between cytosine and guanine are shown here. Two-stranded DNA. Remember, DNA has 2 strands that fit together something like a zipper. The teeth are the nitrogenous bases, but why do they stick together? Replication quiz. What are differences between DNA and RNA? How called monomers of nucleic acids? Describe the structure of nucleotides. Explain the complementary rule to create the complementary strand of DNA. Thank you.